what were your controls? This really is one of the fundamental questions of experimental biology that are not trivial and really have to be thought through very carefully. Effectively, a control is subtracting or adding a critical variable. Negative control is maintaining all the other variables constant, but removing the one key variable that you want to test in your experiment.
So negative control is where that outcome is negative. So it's the absence of something, it's the lack of some effect. So if you think about a drug trial where you have a drug of interest that you're looking to see if there's an effect, you don't know what the outcome is going to be. A negative control for that would be the placebo. So you're administering something that you know should have no effect whatsoever. And that lack of effect is the negative control.
A positive control is an example of adding another variable, in this case likely a known variable that you would expect to produce a particular effect.
In our experiment, our positive control was a cell line that was expressing both HER2 and HER3, the regular HER2 amplified breast cancer cells. So those cells were our positive control because they were generating tumors. So they were giving us the outcome that we were expecting for those cells, were telling us that our experiment was being performed properly.
So to me, very generally, when someone says, this is my control, it means this is a condition where I have an expected result. And if there's a deviation from that result, that tells me that there's some problem, some aberration with the experiment. And without doing controls to check for those possibilities, your data are a lot more difficult to interpret.
Imagine we only put the knockout HER3 cells and we waited for months to see if the tumors develop. And let's say that after 1 year, there was no tumor development. So we would have concluded that those HER3 knockout cells were not able to produce tumors. Just because we didn't have our proper control, that we will have said that something happened during injection or for whatever reason, the mice were not developing tumors. So it's very important to have a positive control because that will allow you to make the proper conclusion with the cells that you are testing.
These controls are also useful to see if there are any other confounding variables that may be affecting the results. In those cases, one often then has to search for what the possible source of that experimental result might be. That is often then a search through all the variables of an experiment, to see what might be producing a particular outcome, or in many cases, the lack of a particular outcome.
I think the really main purpose of a control is to help you interpret the data. If you lack some key experiment to tell you how to interpret it, that's what you really need the control for.